All right, hey everybody. We're going to take a look at Chapter 5. We're going to start with Part 1. And with Chapter 5, we're going to be looking at eukaryotic organisms. So we've looked at the prokaryotes in Chapter 5. And so now we're going to take a closer look in detail at the eukaryotes. So we kind of went over the differences in Chapter 1, and now we're going to go into more detail here in Chapter 5. Okay, so a eukaryotic cell, starting out, is defined as a cell that differs from a prokaryotic cell chiefly by having a nuclear membrane. We'll talk about that. And membrane-bound subcellular organelles. And then this last one here we're not going to focus on as much, but it is part of the definition. It's called mitotic cell division. If you've taken general biology, you studied mitosis and meiosis, and that's in reference to mitosis. But we're really going to focus on the first two, the membrane and the membrane-bound organelles, or the membrane-bound subcellular organelles as they're called here. So what I want you to know about eukaryotes starting out, less about the technical definition. I'm not going to make you recite that. I'm not really worried that you can recite this in its exact sentencing here. But what I want you to focus on is the two differences as they're stated here. So there's lots and lots of eukaryotes, and there's lots and lots of prokaryotes, and they can all be very, very different. Eukaryotes are all very different from each other, and prokaryotes can all be very different. But from a very generalized perspective of what are the general things that we see as a fundamental difference, we see that in eukaryotes, their DNA is always surrounded by a membrane. And we refer to this membrane as a quote-unquote true nucleus. And then here they define it as a well-defined nucleus, but we like to call it a true nucleus. In addition to that, they have internal levels of organization called cellular organelles. The term actually derives from the sense that they are similar to organs of our body. So if you think of humans and want an analogy, or just any animal really, but higher level animals like humans, if you want to kind of make an analogy between humans and eukaryotic cells, just to kind of give us a perspective, if we think of ourself as the cell, organelles would be kind of like organs. And so the idea is that within a cell you have these distinct little components that all serve their own function to the overall whole, kind of like our organs give us functions that serve the overall whole, right? Our liver does one thing, our kidneys do another, our heart does another. It's kind of like how organelles function in a cell. In prokaryotes, we don't really see this. We see kind of one cell in which everything takes place within the confines of that cell. We don't see distinct little units that all serve a better purpose. So there's a very, very dramatic difference between these two cell types, and it really comes down to the organization within the cell. Now, when we talk about eukaryotes, these are the organisms that you're most familiar with, whether you realize that they're a eukaryote or not. All plants, all animals, fungi, and then protists, which we've talked about a little bit, are the algae and protozoa. These also are eukaryotes. So if you can see it with the naked eye, it's a eukaryote. And these are typically the plants and the animals and a lot of fungi. Protists are single celled, as we'll describe down here, but anything you can see with the naked eye is a eukaryote. And this line here, or as it states right here, but this line here attributes to that is that they can come in both single and multicellular form. So when we see it with the naked eye, that means it's a multicellular form. Individual cells are too small to be seen with the naked eye. So the only time you actually see an organism with the naked eye is when you actually have many cells grouped together, meaning you have a multicellular organism. And that only exists in a eukaryotic form. There are no multicellular prokaryotes. You do have colonies and you do have biofilms that become visible, but they still operate as individual cells within the whole. Now, it is true that some eukaryotes are microscopic. So if you see, or if you're looking at something under the microscope, it could either be a prokaryote or a eukaryote. And we'll describe more differences, but microscopic could be both. Something macroscopic, visible with the naked eye, has to be a eukaryote. There maybe is one exception in the whole world that I'm aware of, but you know, this is pretty much true regardless of the example. Okay, so, and just to clarify one more thing, the protista are defined as the single-celled eukaryotic organisms. They are the more common single-celled or microscopic forms of eukaryote. Fungi, as we described, can come in both micro and macroscopic forms. So they can be either, and we'll talk about more of those in the second part of Chapter 5. A few more differences here. On the microscopic level, a eukaryotic cell typically is 10 times bigger than a prokaryotic cell on average. So even though on a microscopic level they're both very small to us, anything that we can't see with the naked eye generally is considered microscopic. On that level, though, there's a difference at size when it comes to the microscopic comparison. So they're about 10 times bigger than a prokaryote, eukaryotes that is. And we also see that they have more advanced features that we'll get into with this first section of Chapter 5. We'll talk about a cytoskeleton. This is a feature that really allows cells to group together and is really a necessary feature for a multicellular organism to form. So we'll talk about that. We see some advanced motility features. We'll look at flagella in a eukaryotic form, and we'll see how they're similar but much more advanced. And then we'll talk about the membrane-bound organelles I mentioned, plus some other things. So these are just a few things in reference to what we're getting ready to talk about here. Okay, so one of the things I want to do is just have you make a quick drawing to compare the difference between a eukaryote and a prokaryote. So let's go, I'm going to go here to my paint document. You can pull out a scrap piece of paper or do it right there in your lecture guide. But what we want to look at here is just a basic stick-figure drawing between a eukaryote and a prokaryote, and then focus on things like the size and the internal structures that we would find in just the eukaryotes, and then those that we would find in both. And generally speaking, if we find it in a prokaryote, we'll also find it in a eukaryote. However, if we find it in a eukaryote, we may not necessarily find it in a prokaryote. Hopefully that makes sense. Okay, let me start with my labels. Over here I'm going to do my eukaryote, and over here I'm going to do my prokaryote. Now, when I draw a eukaryote, I'm going to draw it significantly larger than my prokaryote. And this could be any shape. I'm just kind of going with the general box type shape here. That's about the size comparison. About a 10 to 1. That may not quite be 10 to 1. But that's kind of the general difference is fairly significant just upon a basic observation. You would immediately realize, oh wow, that's significantly bigger. So whether it's exactly 10 to 1 or not isn't important as long as you recognize one is much bigger. Now, going back to our list here, how much larger? 10 to 1. Now, what internal structures would we find in both cell types? So, one, we're going to find a cell membrane. And a cell membrane is a part of the cell that separates the internal and the outside world, the internal cell and the outside world. So a cell membrane is a feature that we will find in both types of organisms. And in fact, a cell membrane is really one of the defining features of all life forms. So a cell membrane is something that we would find in both. And you know what, let me just scoot this over a little bit. What I'm going to do is I'm going to put my features common to both here in the middle, and then I'll put only those on the eukaryotic side over here. So a cell membrane would be found in both types of organisms. We'll label this as common to both. Alright. Just scoot this over a little bit more. Oops, sorry. I know this is painful to watch. I apologize. Well, how about we just trash that? Right, I'm going to get it together here. Common to both. Okay, thanks for being patient. Alright, so another feature that is common to both, and actually let me just draw a line, one more thing here to clarify what we're talking about. Cell membrane, cell membrane. Another feature that is common to both are these units called ribosomes. And we talked about ribosomes back in Chapter 4, and we mentioned that these are structures that synthesize protein. Okay, now I'm going to draw a line to these here in a minute when I add some more features to the eukaryote. So for now, I'll leave those blank because they're structured a little differently. They're found in both, but there's a difference as to kind of where they're found. So for now, I'll leave them blank over here in the eukaryote. In a prokaryote, they're more or less just floating around the cell kind of anywhere within this internal space. And they don't, from our vantage point here, they wouldn't look much more than anything like little specks. So those are the ribosomes. Another feature that we would find in both is what's called the cell cytoplasm. Cytoplasm, this is going to be tough to point to, because cytoplasm is the internal fluid of the cell. So this is just pretty much all the liquidy stuff, so to speak, within the internal compartment of the cell. So in a eukaryote, we'll find that just anywhere within the inside, and the same is true of a prokaryote. So the cytoplasm would just be the liquid inside. And then one more feature that we're going to talk about that would be common to both would be the genetic information. And by genetic information, we mean we need DNA and RNA. So let's clarify that here. Genetic information involves DNA and RNA. Okay. And I'll draw a line to that in the eukaryote here in just a second. And for now, let me go ahead and fill this in. In a prokaryote, the DNA is this big kind of long strand that's kind of coiled up in one part of the cell over here, kind of like that. So we would point a line to that over here like that. Okay. Now let's go back to the eukaryote and let's focus on some features that are common or that are unique just to the eukaryote. And then we'll kind of connect some of these lines that I haven't drawn over here. So one feature that we find in a eukaryote that we don't find in a prokaryote, as we mentioned earlier, is the nucleus. And this is where I said earlier we could call this a quote-unquote true nucleus. And I don't know that it's really necessary to get into why they call it that. That would take time, and it doesn't really help, I don't think. So there's a reason they call it a quote-unquote true nucleus, but for now, just recognize that that's the term. What this really means is that we have our DNA within the cell separated by its own membrane. So let me go ahead and draw the DNA here. So here's our DNA, kind of like we saw in the prokaryote, all kind of coiled tightly. But the difference is we actually have a membrane surrounding it that separates it from the rest of the cell. So if you look over here at the prokaryote, the DNA is just kind of sitting within the membrane, floating around the cytoplasm. Over here in the eukaryote, our DNA is separated by this membrane. So here's the DNA itself inside the nucleus, and then here is the nucleus itself, this membrane around it. Okay. You might think, well, who cares? What's the difference? Well, you can kind of think of like the DNA of the cell as kind of like the brain of a human. It's more or less the most important part, and it also is the central area where things generate from. Kind of like everything you do originates in your brain one way or another. I mean, you could probably argue that, but you know what I mean. The nucleus is similar in that everything kind of generates from the nucleus outward. Okay, so it's kind of like the brain of the cell. So having more separation and having more protection is pretty significant, and it's kind of like the better off, more protection your brain has, the more advanced features your brain has, the more well off you're going to be as a species as a whole. Kind of similar in that sense. So that's a pretty big deal. Another feature that is common, or that is unique just to eukaryotes, is a part of the cell called Endoplasmic Reticulum, and this is often abbreviated as ER. Now, here in a bit, we'll talk about there's actually two different types, but for now, I'm going to kind of keep this somewhat simple and move on here pretty quick. Endoplasmic reticulum branches off from the nucleus, and it's kind of this folded membrane-like structure that branches off usually from kind of one side of the nucleus like that. It can't actually surround it in certain cells, but we'll just kind of keep it simple. And you can see this kind of irregular shaped membrane branching off the nucleus, and that, in a very simplified explanation, is the endoplasmic reticulum. Now, in one part of this is where we will find ribosomes embedded within the endoplasmic reticulum. And here in a bit, we'll describe this as being what's called the rough endoplasmic reticulum. If you took general biology, you may already be familiar with that. But at least in one part of this, we find the ribosomes that we mentioned earlier embedded within here. So here's where the ribosomes are located. And that's kind of why I waited to draw that arrow because they are specifically within that area. There are some that can be floating around the cell, but this is where most of them are found. It's kind of the main center for the ribosomes in the cell. So you see here already a difference, whereas in a prokaryote, DNA is floating, ribosomes are floating, everything is just kind of scattered. In a eukaryote, we see structure. DNA is housed within the nucleus, ribosomes are structured within the endoplasmic reticulum, and we see a more advanced organization, and that's pretty significant. Let's go for a few more features here. Another one that is unique only to eukaryotes is called a Golgi Apparatus. Golgi Apparatus is another folded membrane-like structure that's going to be found separate from the endoplasmic reticulum. So it's usually going to be located just off to the side of that. Get that a little thinner. And it usually consists of a series of flattened sacs kind of stacked on top of each other, kind of like this. And that is called the Golgi Apparatus. We'll talk about what that does here in a bit. For now, we'll just keep it to the drawing. A couple more that's found in all eukaryotic cells. We'll just do one more, and that is the mitochondria. Mitochondria, you may be familiar with, often referred to as the powerhouse of the cell. We'll talk about why that is. But the powerhouse of the cell generates energy, and mitochondria often have kind of a bean-looking shape to them, maybe kind of like this. And there can be many of them floating around the cell. Doesn't have to be just one. So I'll just draw a couple there, and these are the mitochondria here. All right. So when we look at a eukaryotic cell versus a prokaryotic cell, we automatically see a significant difference in terms of the internal organization. Over here we see lots and lots of structures built within the cell, and these collectively, by the way, are referred to as organelles. Let me just make that label over here. These all can be referred to collectively as cellular organelles, or we'll just call them organelles. So these are kind of like the quote-unquote organs of the cell. They have a structure, and they help the cell achieve a higher level of functionality. Over here on the prokaryotic side, we see everything just kind of loosely scattered about in the cell. We don't see a lot of organization, and so what we end up finding is that the difference translates to a higher level of functionality on the macroscopic level. Both the micro and the macroscopic level. Okay, so when we look at how multicellular eukaryotes come about, one of the main reasons is because the internal organization allows for a greater level of macroscopic organization. Okay, so going back and looking at that list here, you should be able to answer these questions here. Now, we didn't cover all of them, but we'll come back to some of the other ones here, or we'll cover some of the ones I didn't talk about as we go forward. I don't want to spend too much time, more than I already have, talking about that. Okay, I talked about cell membranes. I talked about that back in chapter 4. I just talked about it there. I want to just reemphasize these are found in both types of organisms. They're a fundamental feature of all life forms. Here I just want to add a little more detail that I omitted in chapter 4. So when we look at cell membranes, this is again common to all life forms. There's really not a significant difference between a cell membrane of a eukaryote and a prokaryote. There are differences, but for our purposes of this class and this kind of level of biology, it's okay to think of them as being more or less the same. They in a lot of ways they're very, very similar, not identical, but as far as functionally speaking, almost the same. So a membrane is what separates the cell from the outside and the inside of the cell, and they're composed of a type of molecule, a specific type of lipid or fat called a phospholipid. So this is a very specific type of fat molecule. A phospholipid looks like this over here. Individually speaking, they look like this. You have two components to it. You have what's called a head and a tail. And the head is said to be polar, and the tail and actually two tails are said to be non-polar. The difference is that polar reacts with water, and it means it has a charge. In this case, it's usually a negative charge, but sometimes that can mean it's positive. So polar basically means it is slightly charged, either in a negative or positive sense. You can think of like a polarizing person is someone who kind of attracts attention to themselves or is somewhat charismatic. They're not neutral. They're not bland. They're kind of on one side or the other of being good or bad. So that's one way to think about it. Nonpolar is neutral. It means you have no charge. So if someone said you had a non-polarizing personality, that would be meaning that you're kind of bland and non-exciting, no charge to your energy. So in this case, we're looking at the tail as having no charge. So what you actually have in this molecule is two opposing charges, one that's actually charged and one that's not charged at all. And what this actually does is it generates an ability for the cell to create this boundary around itself with these opposing features. Now this is somewhat random because there's lots of ways you could create a membrane. So bear with me in the sense that this is just the way life works. There could be other ways, and I'm just kind of trying to explain it within the way that we actually see life working. So it's not to say that it couldn't have come about other ways. But the way it actually does work is that we see that we have the phospholipids, they arrange themselves in two layers. So we have what's called a phospholipid bilayer, and that's listed right here. This is just a very, very basic overview of a cell membrane. So a phospholipid bilayer means we have two layers of phospholipids. So here you can see an individual phospholipid, and we see the head and the tail. And on the other side, we see another phospholipid with the head and the tail, but they're actually pointing in opposite directions. So the heads on the phospholipid bilayer are going to face to the outside and the inside, and then the tails on both sides are going to point towards the middle. And what this does is it creates in a scenario where the inside of the membrane, where all these tails are, they're actually said to be hydrophobic. And because they have no charge, what it ultimately means, and I'm skipping past lots of detail here, so bear with me, but what it ultimately means, what's important, is that means that that part of the membrane cannot interact with water in a way that water can stay within this area. So it means that this area basically is devoid of water, and that's what the term hydrophobic means. Again, I skipped a lot of reasons as to why. There's a lot of chemistry that you're going to have to know in order for that to really make sense. If you want to go look that up, I'd be more than welcome to do that. But in a nutshell, it means that there's a water-free zone, hydrophobic, meaning water-fearing. So this part of the cell basically creates a water-devoid area where it creates kind of this empty space that things generally don't go into. Most things interact with water, and so anything that interacts with water is not going to be found in this area. So you kind of have a dead zone. It's kind of like if you had a double-layered fence or a double-layered wall, this would be kind of like the space in between where you really wouldn't find too much happening. And the heads is that kind of like the bricks or the concrete itself. Now the head on each side is attracted to water. So what that means is that water can more or less come all the way up to the boundary of those heads, but it can't go past it, at least not in a conventional sense. There's more to that, but I don't want to get into too much detail. So water is basically stuck on one side of the cell and on the other side of the cell. We have a dead zone here in the middle. So what this does is it creates a very distinct boundary between the inside and the outside of the cell. And just one last little point here. Water is essential to life as we know it. So we have to have water in a cell, inside and typically outside to a certain extent. And so this creates a very efficient way to keep water inside and to allow water to surround the cell on the outside. If not pure water, some water-like solution like blood, for example, or some of your extracellular fluid, which is mostly water, your plasma, and mostly parts of your body that are mostly water. It could be some kind of body fluid. But we see that kind of distinct layering here. Now over here is a more advanced drawing of that. We see the phospholipid heads and tails. What we also see in a membrane are little channels and markers and lots of things embedded within and attached to, sticking out of the membrane. And those are some of the things that we're really not going to get into here. That's a topic for general biology. There's lots of little features. Well, I'll mention a few of those here in a second, but not lots of detail. And this is just another figure showing kind of a three-dimensional cross-section of a cell. So this is obviously a flat, very, very kind of basic two-dimensional. This is more of a three-dimensional showing the structure of the phospholipids there. So that's the idea of a membrane. Here's talking about the phospholipid heads. The polar heads face out, the non-polar tails face in, and that's what generates your boundary between the in and outside of the cell. So just a little bit about some of those things I mentioned embedded within the cell. You can think of phospholipids as like the base of the cell, or the base of the membrane of theirs. One of the things that really makes the membrane interactive are the proteins within it. So the proteins, I like to think of them as the quote-unquote gatekeepers, or you can think of them as like channels, kind of maybe like highways in and out of the cell. And what the proteins do is allow the cell to have an active and interactive component with the rest of the world around it, also with other cells within, say, a multicellular organism. Proteins, for example, are what, is a random example, what allow your heart to interact with your brain. Your brain is made up of membrane, phospholipid membrane. Your heart is made up of a phospholipid membrane. But without the proteins, there wouldn't be any way to actually connect those two things. They'd just be these inert little membranes. So proteins are kind of like the active and interactive components that make up the actual key aspects of a cell. So as some examples, what do proteins do? They can allow transportation in and out of the cell. That's where you can think of them as almost like gatekeepers or little highways in and out of the cell. They also allow for attachment. Go back here real quick looking at this. You can see these little structures here, like a glycoprotein, or these little branch-looking structures here. Some of these might actually allow the cell to grip onto something and actually latch on as an attachment feature. They also allow for communication and signaling. Going back here, some of these longer alpha helix type proteins can actually act as a way in which a molecule can land on one side and actually send a signal all the way through the membrane and have it reach the other side. Almost like a wire going from one house to another, almost like your phone lines. Remember when we used to use phone lines? It's kind of like that, where you can actually send a signal from one side of a house to another house, or from one house to another house, or one part of the country even to another, through a physical wire. That's kind of like how communication might take place. Identification, another key feature. Some of these types of glycoproteins actually act as like the equivalent of a facial feature. The cells around them can actually recognize and characterize a cell based on these features that actually give it some kind of characterization to it. I like to think of these as like facial features. They allow cells to identify each other. It's actually something important. For now, we'll just leave it at that. But when we come back to I'll come back to this later when we talk about the immune system. The immune system is largely predicated on the ability of our body to recognize what we refer to as self, our own body, versus non-self, which is everything considered foreign. So our body is largely creating an immune reaction based on the determination of what's self and non-self. So that the idea is we want to attack things that are foreign and not attack our own body that's considered self. Well, the way that our body begins that process is by recognizing cell surface markers, these identification markers, and they can actually tell the difference between our own surface markers and those of foreign cells or foreign entities. And that's based on the unique characteristics of those cell surface markers. They are like a face where they're kind of similar, but each one is just a little bit different. And that's what allows the body to recognize self versus non-self. And then finally, some of the metabolic activities within a cell can actually take place within the membrane, and then others can take place within the cell. So those are some of the features of a membrane. Here's a figure showing some of those different activities. So transport, enzymatic activity, signal transduction like communication. A molecule hits on one side, passes a signal inside. Recognition, a feature here allows another cell to recognize. Attachment, adhesion, two cells can join together. And here's another type of attachment where a cell can attach to a different type of structure altogether. So some of those same things we just talked about there with a few illustrations for those.